Yes, I'm Ewa Szczurek, and I will talk about machine learning in medicine, which is the research focus of my lab at the moment. But before I start, I want to thank the organizers of this event for bringing us together and having the opportunity to present our research. And I look forward to discussions at the table. Right. So what do we do in the lab? We mostly focus on method development, and currently we work with machine learning, and we focus on probabilistic graphical models and deep learning, and we do some general statistical data analysis. But this method development is always at the service of what we think is an important medical cause. And these medical topics include cancer, antimicrobial peptides, and COVID-19. And I will try to present some more details of this, but if you will be interested in knowing a bit more, please visit our website. The link is shown here below. So here you see a very broad overview of our research topics, and we always somehow are around the question of how to model and understand different things. And these different topics include sensitivity of cancer cells to drugs, generation of antimicrobial peptides, analysis of medical images, modeling tumor evolution and interaction of the tumor with its microenvironment. Very recently, also COVID-19 epidemiology, and a lot of, somehow there's a large bucket of high-dimensional experimental medical data. Oops, sorry. All right, so what are the particular examples of methods that we developed in the area of deep learning? So in 2019, we published so-called ARA-CNN, which is our convolutional neural network model that also is trained using active learning. And this was a model specifically developed for classification of histopathological cancer tissues. We also now published or posted on bioRxiv DEARS, which is an autoencoder-based and recommender system-based model for drug sensitivity prediction. And finally, we also have developed HIDRAMP, which is a conditional variational autoencoder for generation of antimicrobial peptides. So all these models are very highly flexible. They have multiple, like really many parameters. And as such, they can be, and if you have enough data, they can be trained to be really highly accurate and really highly predictive. And research in this field is just amazingly rapid and very exciting. But there is one, there are actually many caveats of this approach, and one of them is that these methods are quite difficult to interpret. So we also have this other sort of leg in the lab. We develop probabilistic graphical models, and to me, they are actually very different to these deep learning approaches, because they are models that are typically relatively smaller. And then you actually interpret each random variable in this model in a very specific way. So each random variable corresponds to some entity in the model system. And you are specifically designing yourself what are the conditional probability distributions that can describe relations between these variables best. And generally, I think these models allow more precise description and more in-depth understanding of what you're actually modeling. So here I'm showing two graphical representations of such models that we developed in the lab. One of them is called Cactus. It was published in Genome Medicine this year. And this is actually a tool that combines data from single-cell RNA sequencing with models of tumor evolution inferred from whole exome tumor sequencing. And we learn parameters of that model using Gibbs sampler. Another example is Celloscope, which is a model for for spatial transcriptomics data. And in this model, we learn parameters using Metropolis Hastings within Gibbs. So going more towards the end of the presentation, I want to show you an example PhD thesis, which, so just to see how papers from such thesis can look like. And this thesis is focusing on drug sensitivity prediction, and the PhD student here is Krzysztof Koras. And what you're seeing here is the first paper of Krzysztof, which he published last year. And his first project focused on feature selection strategies for drug sensitivity prediction. So in this work, he has trained multiple traditional machine learning models for predicting of sensitivity of different individual drugs on cancer cell lines. And I think it was a very nice first project because he learned a lot about what this data actually is, what are these drugs, and what are the features of the cell lines, what are the features of the drugs, and how to model this as a first approach. And in his second project, which he posted earlier this year, he already developed a new approach, which is a multitask learning and a recommender-based system approach, where he's modeling all drugs and all cell lines at the same time. We have information about 74 drugs and their inhibition strengths for 294 cell lines, kinases. And then we have 922 cancer cell lines and 241 features of these cell lines. And now in a very traditional recommender system way to work with such data would be to use linear projections of this site information data into lower dimensional space, and then to use dot product to transform these linearly projected data into a matrix, which will give us prediction here of drug sensitivity, sensitivity of cancer cell lines to different drugs. And this sensitivity is measured here using a so-called AUC or IC50. So this is, let's say, inherently a linear approach, and we extended this model using nonlinear models, and in particular deep learning, specifically autoencoders. So for the projection into lower dimensional space, we use drug autoencoder and cell line autoencoder, and now hidden representations of the drug data and the cell line data are concatenated and then fed into a forward network to predict the response of the cell lines to the drugs. So here the I hat is either AUC or IC50. And now his final project is to even further extend that model and now work not with inhibition profiles as the input for the drugs, but actually to take textual representation of the compound chemical structure, which is called SMILES, as input, and then again use a smart autoencoder to actually generate inhibition profiles for each such drug, reconstruct its SMILES representation, and also to predict the response of that drug to cancer cell lines. And now on the level of application of that model, if this is successful, we are currently working on this, we will be able to actually very largely expand the space of modeled drugs, because for many drugs there exists the SMILES representation, but not inhibitory profiles and not their drug response data. And finally, we also are working currently on much more detailed than normally modeling of the latent space representation. We want to have a generative probabilistic graphical model on this latent space, and in such a way we probably will be actually even able to generate new candidate sequences of these SMILE sequences for drugs, which could have very good inhibitory profiles and very strong response of cell lines to such a generated drug. This is our, let's say, hope for this final project, which I find very exciting because it marries this deep learning way of working and the probabilistic graphical model way of working. So I wouldn't actually explain our research fully if I wouldn't introduce you to our group. I think that when thinking of a PhD, it's very important to find a good research group where you will feel well. So here is a photo of us on a Zoom meeting. We are working on online now. We have several PhD students and some master students, and I think I really actually appreciate all of them, and I really like my group. I think the people are great. We have a lot of funding. We have a lot of fun collaborators from across the world and also inside Poland. And with this, I actually want to finish. I want to say we have some positions open. If you're interested, please have a look on our website. There are actually really many possible PhD topics that we could undertake. Some examples include machine learning for digital histopathology, probabilistic graphical models for spatial transcriptomics data, autoencoders or probabilistic graphical models for single-cell RNA-seq data, and finally, probabilistic model of the latent space in autoencoders. If you're interested in any of this, you can definitely chat with me at the table. You can email me. And thank you very much for your attention. And I can answer questions if there aren't any, and if there are any, and if there's still time.